MARC and SRC kinases, structural gatekeepers in cancer signaling. MARC kinases, part of the AMPK-related kinase family, regulate microtubule stability by phosphorylating microtubule-associated proteins, MAPs. This affects cell polarity and division, processes often hijacked in cancer. SRC kinases, non-receptor tyrosine kinases, are activated downstream of receptor tyrosine kinases, RTKs, and integrins. They modulate focal adhesion, cytoskeletal dynamics, and cell motility. Crosstalk between these kinases occurs through shared downstream effectors such as FAK, focal adhesion kinase, activated by SRC and influenced by MARC-mediated cytoskeletal changes, MAPK and PI3K/AKT pathways. Both kinases can modulate these pathways, promoting proliferation and survival. Both MARC and SRC kinases share a conserved kinase domain with an ATP binding pocket, but differ in regulatory domains. Kinase key structural features. Functional role. MARC, kinase domain, UBA domain, Ka1 domain. Microtubule regulation, cell polarity SRC, SH2, SH3, kinase domain. Signal transduction, adhesion, migration, SH2/SH3 domains in SRC mediate protein-protein interactions, crucial for signal specificity. Ka1 domain in MARC helps localize the kinase to specific cellular compartments. These structural motifs are prime targets for drug design. Structure-based drug design uses 3D structural information of proteins to design molecules that bind with high specificity. For MARC and SRC kinases. Potential strategies: ATP competitive inhibitors, bind to the conserved ATP binding site, for example dasatinib for SRC. Allosteric inhibitors, target regulatory domains to modulate activity without competing with ATP. Protein-protein interaction blockers, disrupt SH2/SH3 domain interactions in SRC or Ka1 domain localization in MARC. Structural and therapeutic comparison of MARC and SRC kinases. Comparative overview. See SRC kinase has kinase domain and other domains are SH2, SH3, where else MARC kinase has domains kinase domain and other domains such as UBA domain. Active site ATP binding pocket within kinase domain, ATP binding site regulated by phosphorylation in all these proteins. Cancer associations with SCR are breast, colon, lung, pancreatic cancers, glioblastoma, pancreatic ovarian cancers. Approved inhibitors for SRC are dasatinib, bosutinib, saracatinib, where else for MARC4. Drugs are still in experimental phase. Drug design approach. Structure-based design targeting ATP binding site, high-throughput screening and structural modeling. Visual structures, co-crystal structures with inhibitors available. Structural models highlighting inhibitor binding. This comparative analysis underscores the significance of structural insights in the development of kinase inhibitors for cancer therapy. While SRC kinase has well-characterized inhibitors with clinical applications, MARC kinase remains an emerging target with ongoing research into its structural biology and inhibitor development. Clinical implications. SRC inhibitors like dasatinib and bosutinib are already in clinical use for leukemia and solid tumors. MARC inhibitors are under investigation for neurodegenerative diseases and cancer, with potential to impair tumor cell polarity and metastasis. Combining inhibitors targeting both kinases could offer synergistic effects, especially in metastatic cancers where cytoskeletal remodeling and migration are key. Conclusion: MARC and SRC kinases, though structurally and functionally distinct, converge on critical cancer pathways. Their well-defined structures make them ideal candidates for structure-based drug discovery, offering hope for more precise and effective cancer therapies.